All right, time to go teach. Hello and welcome to this next episode of Tutors, continuing our unit on genetics, and today we're going to be looking at the gametes and the probability that a parent can pass on a certain type of allele. So let's look at our targets of the day. First target of the day is to be able to track alleles between our different generations, because really that inheritance pattern is what genetics is all about. And next, we're going to identify possible genotypic and phenotypic ratios that can come about for different or very specific parental crosses. Each parental cross results in a specific expected genotypic and phenotypic ratio for the offspring. So, here we go. Remember, we have meiosis, which is going to produce our gametes, and if we started off with a homozygous parent, so this is homozygous recessive for being green in the P, then what we would anticipate is, as these homologous chromosomes replicate, and then they would be separated during meiosis, we would only be able to have one specific type of gamete. Each of the gametes would have a recessive A allele, which is for the P color. So that is why our P's would look green here, and all of its gametes are going to have the allele causing the green color P's. But if we started off with a heterozygote, where it has both the dominant allele and the recessive allele, when our chromosomes replicate in the S phase of interphase, we still have the dominant A allele on our first chromosome here, and we have the recessive A allele on our second chromosome. And even when crossing over occurs during meiosis, we still end up with only two different types of gametes for this specific gene. We only have it passing on the dominant A allele or it passing on the recessive A allele. And that means we can look at it as the options that each parent can pass on. If we had a homozygous parent for the dominant allele, it can pass on either the dominant A allele or it can pass on the other dominant A allele. Those are the only two alleles that it can pass because they're the only two alleles that it has. And likewise, our homozygous recessive individual, well, that first A allele, the first little A allele, it can either get paired up with our first option, or it could end up getting paired up with our second option. The last little A allele could also be paired with our first dominant allele with our first pairing, or it could end up getting paired with the second dominant allele. Either way, each of the offspring that could be produced from this pair would end up being heterozygous, having both the dominant allele and the recessive allele. These are the different combinations that it could have occurred in, and as it results in that they all would be exactly the same in regards to the genotype and the phenotype for this specific gene. That leads us to this tool that we use that many of you have heard about. It's called a Punnett Square. How a Punnett Square works is exactly what we just did, except it's going to show it in a nice orderly fashion. Each parent has two of each gene, one on each homologous chromosome. And if we look, it can pass on either one of these different genes. It can either pass on this first allele, or it can pass on the second one, depending. And likewise, our other parent can either pass on this first allele, or it can pass on the second. Those are the only options that these parents can pass on because those are the only alleles that these parents have for each of these genes. Now, we can just figure out our combinations. We can just bring our alleles across from the side, and we'll drop our alleles down from the top to again show that all we can end up with a combination of one parent being homozygous dominant and the other parent being homozygous recessive, all of the offspring are going to end up being heterozygous. We have a 100% genotypic frequency of being heterozygous. That is a 1-to-0 ratio. We also have a 100% chance that they would all appear dominant. Again, that's a 1-to-0 ratio for being dominant. Some people look at Punnett squares and think that they have to be a 2-by-2 grid. That is not the case. Notice that we have only one actual option that this homozygous dominant parent can pass. It can only pass on the dominant A allele. And likewise, we only have one option that the homozygous recessive parent can pass. It can only pass on the recessive A allele. And that means that we didn't have to draw a 2-by-2. We could have only shown it using a 1-by-1 grid, and that reveals all the options because we have 100% that would be a genotype of being heterozygous and the phenotype of appearing with those yellow P's. So now let's go to the F2 generation where we cross two heterozygotes together. We have our heterozygote, both sides. These are our parents. They came from the F1 generation, so this is going to produce the F2 generation. And again, we're looking at the options. We could either pass on the dominant allele, or this one can pass on the recessive allele. It has two different options. Likewise, this far parent can also pass on either the dominant allele or the recessive allele. And just like last time, we're going to fill in our Punnett square, bringing our alleles across and our alleles down. And now we have our F2 generation, which shows that 50% of these are heterozygous. So we can see that these 50% are heterozygous. And we can see that 25% are going to be homozygous dominant, and 25% are going to be homozygous recessive. Those are the expected genotypes that we would produce from our F2 generation. Now, that doesn't mean that we would actually end up with exactly 25% that would be homozygous dominant, and 25% that would be homozygous recessive, and 50% that would be heterozygous. It's the expected genotypic ratio. That might not be how it actually turns out, but is what we would expect for our offspring to be. The phenotypes, we would anticipate that 75% of the offspring would appear yellow, and we would anticipate that 25% would appear green based upon their genotype. But again, that might not be exactly how it shows up. We might end up with some variation. And if we have only a few offspring, we might actually be very different from our expected phenotypic ratios. But it's based upon the fact that if we had a lot of offspring, we would expect them to fall pretty closely with the 75% that would be yellow and the 25% that would be green peas. So those are what we call monohybrid crosses. They're where we took only one trait that we were looking at. In that case, we were looking at the pea color itself. Green peas, which is recessive, and yellow peas, which is dominant. And then we were able to predict the outcome of the parental crosses. We could then analyze potential outcomes, not necessarily what would happen, but what we would anticipate would happen. It's very similar to if I were to flip a coin. If I were to flip a coin, 50% of the time I would anticipate I would get heads, and 50% of the time I would anticipate I would get tails. That does not mean that once I flip it and I get heads, that the next flip I would get tails. It just means that if I were to flip a coin a bunch of times, I would anticipate that I would get about a 50% and 50% outcome. So now that we've seen this, what are the ratios, both of the genotype and a phenotype, for each of these crosses? The heterozygous crossed with a homozygous recessive, a heterozygous crossed with a homozygous dominant, and lastly, heterozygous crossed with a heterozygous. You can use whatever letters you wish to represent the alleles, but I recommend using a letter that is very easy to tell the difference between the capital and the lowercase. That's why I was using A's on the past example. So, in a second, I will show the answers for these crosses. All right, now we're going to look at the answers. So for the first one, we're looking at the heterozygous crossed with a homozygous recessive. That would be this. We have our heterozygote crossed with our homozygous recessive. And you can see I didn't actually do a full 2x2 Punnett square because I didn't need to. The homozygous recessive can only pass on the recessive allele, so I ended up with a 2x1 cross. And we can see the genotypic ratio that we would anticipate is a 1-to-1 ratio here. 1 being heterozygous and 1 being homozygous recessive. That is a 50% split. If we were to look at this, we would anticipate that half of the offspring would end up being heterozygous and the other half of the offspring would end up being homozygous recessive. And that means phenotypically, we would anticipate again that we would have a 1-to-1 ratio where 50% would appear for having the dominant trait and 50% would appear to have the recessive trait. What about the next question? Well, that was a heterozygote crossed with a homozygous dominant. Again, using only a 2x1 square because the homozygous dominant can only pass on the dominant allele. The genotypes, well, it ends up being a 1-to-1 ratio again, where we have 50% that would be homozygous dominant and 50% that would be heterozygous. In the phenotype, we would anticipate that it would be a 1-to-0 ratio, where 100% of our offspring would have the dominant trait. There's 0% chance that they could appear with the recessive trait because we don't have the option for an individual being homozygous recessive. And that's the only way that you're going to end up with a recessive trait showing up is if we happen to have an offspring that has a genotype that is homozygous recessive. So this is a 100% chance that the dominant trait is what's going to be shown in the offspring. And now our last one, which was a heterozygote crossed with a heterozygote, which is exactly what our example was with our yellow peas. So you can see, again, that this is a 2x2 square that we would have. This shows us our genotypic ratio would be a 1-to-2-to-1 ratio. That would be 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. Which leads us to anticipate that 25% of our offspring would end up with the homozygous dominant genotype. 50% of our offspring would end up with the heterozygous genotype, and 25% of our offspring would end up with the homozygous recessive genotype. The phenotype, on the other hand, that would be a 3-to-1 ratio, where 75% would appear to have the dominant trait, and 25% would appear to have the recessive trait. And those are all expected ratios. They might not be what actually turns up, but it's what we would anticipate if we were to cross these different parents. So in summary of all this, alleles, they're passed down from the parents to the offspring, and this is called inheritance, and that's what we look at when we're looking at genetics. It's all based upon the alleles that the parents have. The parents cannot pass on alleles that they don't have, so it's all dependent upon those and what gametes those parents can make. From that, we can calculate the expected genotypic and phenotypic frequencies that they would have for their offspring. And this gives us an idea of what % chance they have of passing on a specific trait to their offspring, whether or not they might have a 75% chance of passing on a trait, or a 25% chance, or even a 0% chance of passing on a specific trait to their offspring based upon the parents' genotypes. But those are all what we would anticipate and not necessarily exact figures. They're just a clue on what we could end up with, not necessarily exactly what we would end up with. The only way to find that out is by actually performing the cross and having the parents mate and seeing what the offspring actually turns out to be. And that is it for this time. Until next time, be awesome, stay awesome.